Assalamu alaikum. I am Dr. Iqra Munir. I am serving as an assistant professor in the University Institute of Medical Laboratory Technology under Faculty of Allied Health Sciences, the University of Lahore. The topic of my today's talk is the introduction about the viruses. When we define virus, what is a virus? Virus is actually an infectious agent that replicates only inside of the living cells of their host. And when we see the spectrum of of viral host, it includes several species of animals and several species of plants and also some species of different bacteria. Now, if we talk about the origin of these viruses, there is no specific agreement on how these viruses have evolved. When we see the history or evolution of these viruses, there is no universal agreement on how these viruses have originated, and there are actually three different evolution theories that are given by different scientists that include the regressive evolution, cellular origin, or their origin as the individual entity. Now, these viruses are actually the most abundant microorganisms on Earth, and they actually outnumber all other microorganisms on the Earth. These viruses are playing a very important role in the evolution of all the hosts that they infect. That includes bacterial species, some archaeal species, and also in eukaryotes. The viruses are actually the obligate intracellular parasites, and they are unable to replicate or multiply outside their host. When we talk about the size of viruses, it ranges between around 0.2 micrometer to 0.3 micrometer, and we usually require electron microscope for visualizing these viral particles. And if we compare the viruses with the other microorganisms, and specifically with the bacteria, they are about 100 times smaller than the bacteria. If we talk about the structure of virus, these viruses actually do not bear any resemblance to the cells. They lack the cytoplasm, they lack nucleus, they lack mitochondria, and also they lack the ribosome, which are the protein synthesizing machinery of the cell. So when they are lacking all these important components that are required for the growth of the virus, they actually hijack the machinery of the host cell. The viruses contain only the parts that are needed to invade the host and also to control the host cells. When we talk about those components that all viruses possess, they include the genetic material that can either be the DNA or it can be the RNA of the virus. And the second important component is the viral protein coat that is protecting the genetic material of the virus. And some viruses, they also possess the envelope. So those viruses which possess the envelope, they are known as enveloped viruses. And those viruses which do not possess the envelope, they are known as non-enveloped or naked viruses. So this diagram is actually showing the genetic material of the virus, the capsid that is surrounding that genetic material, and the envelope that is present on the outer side. The capsid is present in all types of the viruses, and this capsid is actually made up of smaller subunit proteins, which are known as capsomeres, and many capsomeres, when they join together, they make the capsid of the virus. And the capsid along with the nucleic acid is known as nucleocapsid. And the capsid together with the nucleic acid is the nucleocapsid. Some viruses have external covering, which is known as the envelope, and the viruses that are lacking it, they are known as the naked viruses. This is the comparison between the naked and the enveloped virion. At the outermost side, the virus attachment proteins are there, which are helping the virus to attach to the host cell receptors. Now, if we see the function of this viral envelope, it is almost present in all animal virions or animal viruses, and this viral envelope is actually acquired when the virus is replicating inside the host cell, and when it has replicated and when it is leaving the host cell, it actually modifies the cell membrane or the nuclear membrane of the host cell and wrap it around the viral particles, and then it moves outside the host cell, and this is how it acquires the envelope. Outside the envelope, there are some exposed proteins that are known as the spikes, and they are helping the virus in the infectivity. On the outermost side of the envelope, there are spike proteins that are helping the virus to attach to the host cell. Now, if we talk about the overall function of the capsid and envelope, they basically provide the protection to the genome or the nucleic acid when the viruses are present outside the host cell, and they are helping the virus to bind to the host cell receptors and to cause the infection into the host cells. When we talk about the general structure of the viruses, mostly the viruses they are having two types of structures. One is helical symmetry, and the other symmetry is the icosahedral. So all viruses that appear to be rod shaped, they are having the helical symmetry, while those viruses that appear as spherical forms, they are having the icosahedral symmetry. Apart from the helical or icosahedral symmetry, some viruses are unusual, or they are known to be as the complex viruses. They are actually the atypical viruses. They do not exactly resemble to the helical or icosahedral symmetry, and the examples that we have of these complex viruses include the pox virus as well as the bacteriophage. There are many ways to classify the viruses, and when we divide these viruses on the basis of different taxonomic groups, we have different classifications. Number one, the host range of the virus. Number two, the morphological characteristics of the virus. Number three, what are the modes of replication of that virus. Number four, the nature of the disease that they are causing, and their genetic material and the characteristics of their genome. When we divide them on the basis of their host range, we can divide them in three different categories that include the animal viruses, the plant viruses, and the bacteriophages. When we divide the viruses on the basis of their morphological characteristics, and specifically when we talk about the virion structure, we can divide them into three different categories based on the symmetrical patterns, which includes the helical symmetry, icosahedral symmetry, and the complex symmetry. When we divide the viruses based on the composition, we divide them in two categories. Number one, the naked viruses, which are lacking the envelope, and the second one are the enveloped viruses. The viruses are also divided into two different categories based on the mode of replication, and we divide them as lytic viruses and lysogenic viruses. So what are lytic viruses? These viruses are actually that category of virus that replicate inside the host cell and burst the host cell and move out of the cell. While the other viruses include the lysogenic viruses. These are the viruses that live synergistically with their host, and they are also known as the latent viruses. So when we divide the viruses on the basis of nature of disease, we divide them as the viruses that cause meningitis, that cause eye infection, that are infecting the lungs, that those viruses that are causing infection in the gastrointestinal tract. Those viruses, some viruses are causing the infection of the skin. Some viruses are causing sexually transmitted diseases, and some of them are causing infection in the liver, etc. So when we divide the viruses based on the genetic material, we divide them into two major categories, which include the DNA viruses and the RNA viruses. The DNA viruses and RNA viruses are further divided into two different categories that include the single strand or the double strandedness of the viruses. Furthermore, the RNA viruses are also categorized as those viruses which are carrying positive sense genome or negative sense genome. Apart from the characteristic of the genome, some viruses are also carrying the specific enzymes that are required by these viruses that are not present inside the host cell, and those enzymes include the polymerases, replicases, and the reverse transcriptases. The polymerases and the replicases, these enzymes are required by the virus, which are helpful in the replication of the genome of the virus, while reverse transcriptase enzyme is important when the virus has to convert its RNA genome into its DNA genome. Now let's see what are the current basis of viral classification. So this is basically the combination of three different characteristics of the viruses, which include the structure, chemical composition, and also the genetic makeup of the viruses. And based on all these three combinations, we have divided the viruses into three orders, 63 families, and 263 different genera of the viruses. And when we classify these viruses, their family name will always end at the word viridae, and their genus name ends at the word virus. For example, herpes simplex virus type 1. This table is actually showing different medically important viruses which are further categorized into DNA viruses and RNA viruses. Among DNA viruses, we have the example of smallpox virus and hepatitis B virus. And among the RNA viruses, we have several categories, which the example includes the polio virus, HIV virus. Among the RNA viruses, we have different examples, which include polio virus, HIV, hepatitis C virus, coronavirus, and influenza virus. According to Baltimore classification, this is basically a system to classify the viruses based on the genome that virus possesses, its single-strandedness or double-strandedness, and also the way that how viruses are making their proteins from the messenger RNA. This classification was devised by David Baltimore, who is a well-renowned virologist, and he gave this classification in 1971, and he classified these viruses into 7 different categories. In class 1, there are double-stranded DNA viruses, which can directly make their messenger RNA, and further proteins are being made. In the second class, there are the viruses which possess single-stranded DNA as their genome, and there is an intermediate step that includes the formation of double-stranded DNA, and then the proteins are made from the messenger RNA. In the third class, these are the viruses which are possessing double-stranded RNA, and they can make directly their proteins from their genome. In the fourth class, it is the category of single-stranded RNA viruses which are having positive sense genome. So there is an intermediate form in which their single-stranded positive sense RNA genome is converted to negative sense single-stranded RNA genome, which is then converted to the mRNA, and it can make its essential proteins. In the fifth class, it is the category of negative sense single-stranded RNA genome, which can directly produce its messenger RNA, and further proteins are being made. The class 6 viruses convert their single-stranded RNA genome into DNA RNA duplex with the help of reverse transcriptase enzyme, which is then again converted to double-stranded DNA, and further it is converted into messenger RNA. In the 7th class, we categorize those viruses which are having double-stranded DNA and also they possess reverse transcriptase enzyme, which is helpful in the formation of messenger RNA later in the replication stages of the virus. Thank you.